Welcome, everyone. It's my great pleasure to introduce Bridget Carragher today. The opportunity to hear Bridget speak about her own work here at Scripps is rare, and I have been looking forward to this seminar for weeks. As many of you know, Bridget is not only a professor here at Scripps in the newly created Department of Integrative Structural and Computational Biology, but she's also the director of the National Resource for Automated Molecular Microscopy here at TSRI. What you may not know is that Bridget got a bachelor's and master's degree in physics from the University of Witwatersrand in Johannesburg in South Africa, and a PhD in biophysics from the University of Chicago. Her professional career has included various research positions in academia and industry, and in 1992 she accepted a job at the Beckman Institute of Advanced Science and Technology at the University of Illinois in Urbana-Champaign, where she was eventually director of their imaging technology group. And it was at the Beckman Institute that Bridget started to work together with Clint Potter, and the two of them moved to Scripps in 2001 to set up and head the what we now know as NRAM, the National Resource for Automated Molecular Microscopy. So Bridget has been interested and passionate about electron microscopy for her entire career, and her particular goal is to push this technology to its limits using both developments in hardware and software. And a hint of that was already apparent in her first paper as a graduate student with Bob Josephs, where she reported the development of an algorithm that corrected beam-induced movements of samples during electron microscopy. So working together with Clint in the 1990s, she solidified these ideas and evolved these ideas and became a major and highly respected contributor in the field of electron microscopy, which really fundamentally changed. One of her key goals is to automate routine tasks based on an attitude that such tasks are better performed by a computer than by humans. And this philosophy has driven her career in the last 2 decades, and it culminated in the development, together with Clint, of a program that's called Leginon, and that has really revolutionized cryo-EM data collection because it has automated the entire process. It's a completely portable program that these days, it was first, I think, announced in the late 90s or made available in the late 90s. It's now used in many of the best labs throughout the world, both for electron tomography and single particle reconstruction. Clint and Bridget have done many other wonderful things that I don't have time to talk about. And I know it's Bridget's moment to shine tonight, but she doesn't mind that I mentioned Clint too, because the two of them are such an integrative team that it's really impossible to sort out who has done what. Suffice it to say that in the last 10 years, Bridget and Clint have created a truly world-class environment here at Scripps that they like to refer to as the village. And what it does is it brings extraordinary expertise to any EM problem, be it software, hardware, or just structure determination. And they have assembled a group of people that are tremendously knowledgeable. So the users that come here to NRAM are coming not just because they can do EM, they can do so much more. The ideal scenario is for people to come here with a sample and to leave with a structure. And that is something that they have essentially achieved, and it's a remarkable achievement. So how is that possible? It's possible because both of them are extremely dedicated. Bridget, for one, Jack and I were talking about Bridget yesterday, and Jack said, Bridget is an enabler. She takes ordinary people and makes them into extraordinary people and has them do extraordinary things. And so I think that is certainly true, and with that, I will not take away more of Bridget's time. To the audience, I will say prepare yourself to be inspired and motivated. And please give an energetic welcome to our speaker, Bridget Carragher.
Hello, everybody. I'm very honored to be part of this lecture series, which I've successfully avoided for years. Thank you, Aneta, for bringing that to a conclusion. It is very intimidating. And as Aneta kindly introduced for me, this is really on behalf of Clint and I, because we work so closely together. So the focus of the work that we do is really the development of new technologies for automating and streamlining transmission electron microscopy. But the purpose of that technology is really to investigate the dynamics of molecular machines. These are the working engines of biology, and I would say arguably the most fascinating entities you can possibly study with structural biology. So what I'm going to do in this talk is introduce some of the methods to you to start, a very short introduction, just so you know what we're up against. Then I'm going to show you how we've used this technology to solve several macromolecular machines of interest to our collaborators, PIs at Scripps. And then I'm going to end in the last bit of the talk talking about the future, what's becoming possible, what we've envisioned as being possible. And I think it's fair to say we have a very bright future ahead, both for the technology as well as for our goals of investigating the dynamics of these molecular machines. So, first, a brief introduction to what transmission electron microscopy and single particle reconstruction is. So what we start with is bulk sample. These are your particles or your structures floating around in solution. And our goal is to put a drop of that on the end of an electron microscope grid, and then we plunge it into a really good cryogen, and we basically reduce it to a thin, rigid film. So if we look at that thin film in the electron microscope, we pass the electrons all the way through it, because it is transmission electron microscopy, and we produce an image. And if we look at that image, what we see are the particles embedded in their aqueous medium, and there's different views of these particles, and that just happens to be because the particles are in different orientations in solution, and so they're differently related to the electron beam. So if we look at the images, one of the problems with them is that they're very noisy. The signal-to-noise ratio of these images is typically way less than 1, and that's because they're delicate, and if you expose them to electron beam, you're going to damage them. So we're constantly up against the signal-to-noise barrier, and we have to do something about it. So what can we do? We use the power of averaging. There are lots of copies. If all these particles are identical, then many of them will be in the same relative orientation, and we can pick all those out, and we can cut them out of the image, and we can align them relative to each other, and then we can average them together. And so if we can do it with that view, we can do it with all the other views. And then with the Fourier slice projection theorem, we know that if we have many different projection views of a three-dimensional object, we can combine those back to form the three-dimensional structure. And this is one of the first things we did at Scripps, was to demonstrate that we could do something like this in about 24 hours after putting the specimen in the electron microscope. And the resolution that was available, this is about 10 years ago, is such that you can dock high-resolution crystal structures into the structure. So you can see secondary structure, and you can basically dock the structure in and understand how those domains fit together. Resolution in electron microscopy is determined by a number of different factors, several of them being the size of the particle, its symmetry, because that determines how many asymmetric subunits you have, the number of particles you average together, and then, of course, the flexibility and heterogeneity of the particles. So for a rotavirus, this enormous structure, 70 megadalton, you can reconstruct it with these standard methods to a resolution where we can actually start to see all of the backbone of the particle, and we can trace out the chain de novo, and we can see even some of the big side, all the bulky side chains and the bigger side chains. So that's what's possible with this enormous structure. But how about some little guy like DICER that Ian McRae works on? This is a machine that chops up double-stranded RNA into exact bits as part of the RNA silencing pathway. DICER is small, it's flexible, it's much more difficult to work with. And as a result, the reconstructions we get are really these so-called blobology. It's a big blob, but with all kinds of fancy labeling techniques, we can still fit the various domains into that blob and figure out how that machine works. So we can still do something with something that small and flexible. So the difference between these two, of course, this is enormous, 70 megadalton. This is 220 kilodalton. This is 60-fold symmetric. This has no symmetry. This is very rigid. This is very flexible. And so the resolution here is under 5 angstroms, where we can trace a backbone. And the resolution here is only 20 angstroms, where we can dock in domains. And the reason for that is the signal-to-noise problem. If we look at a rotavirus by the electron microscope, that's what it looks like, an obvious structure. Little dicer looks like that, and if we scale them up to the size of what they are, it's easy to see we could align and average something like that. This is mostly noise. It's very difficult to align and average together, and that's what's really limiting our resolution, and I'll come back to that point several times during the talk. So what can we do to make this easier? So when Clint and I arrived at Scripps in 2001, the very first thing we did was sit down and write a proposal to establish the National Resource for Automated Molecular Microscopy. And our overall mission was to develop, test, and apply technology for automating and streamlining all of the processes that are required to go from a specimen to a 3D map and get the structure of molecular machines. And to do that, we had various technology aims in specimen preparation, in automated image acquisition, and in streamlined data processing. And what we wanted to enable was the accessibility of the whole method. We wanted anybody with an interest in getting a structure to be able to do it, not just the experts who'd trained for years. We wanted to do this with much higher throughput because we're very impatient people. And then we wanted to be able to enable the high resolution structure of small, like dicer, asymmetric, and perhaps very heterogeneous particles, because all these dynamic machines, because they are dynamic and they are working machines, tend to be very heterogeneous. And we may need to analyze millions of particles in order to be able to get the structure, so obviously a high level of automation is called for. And we may need to determine very many three-dimensional structures in very many different states if we're going to be looking at dynamics. So automation is called for there. So the real goal then was the investigation of the structure, the function, and the dynamics of molecular machines, not simply the structure. I think structure without exploring into function and dynamics is much less interesting. So what did we build up? This is the sort of current state at NRAM. So we've built up a bunch of technology in specimen preparation. I'll talk a little bit more about that later because this needs a lot more work. We've built this software system called Leginon, which really is the system that once you have the grid in the microscope, just sits there all day doing multi-scale images and pumps out thousands and thousands and thousands of images. Many years ago, we passed our millionth image in Leginon. I have no idea how many we're up to now, 2 million or so, I guess. And then more recently, we built this very streamlined processing pipeline that uses a whole bunch of web pages to provide access to these sophisticated processing packages backed up by a giant database. And this enables you to keep track of these complex processing procedures. So these are some of the structures solved at NRAM over the last few years, some as early as 2003. This was Jerry Joyce looking at one of these DNA origami, and some as recently as a few weeks ago, this brand new, interesting bacterial structure. So I'm going to tell a story about a few of these, just to illustrate what we can do. And appropriately, one of the first stories is a collaboration done with Jack Johnson. Jack is one of our favorite people. He's, as everybody knows, incredibly enthusiastic, and he's really been so supportive of everything we've tried to do here. One of the things we're most grateful to Jack for is he introduced us to Gabe Lander, a graduate student in 2005, and Gabe worked between the two groups for the next several years to look at all kinds of structures, including looking at these infection conduits. So what are these? So the co-evolution of phage and bacteria represents, one of the most fundamental struggles for survival in biology. Evolutionary pressures cause various mutations in the phage, and they evolve, and various mutations in the bacteria, and then these have to co-evolve to fight each other in what is essentially an evolutionary arms race. So the phage themselves are the most abundant particle on our planet, most abundant particle in the biosphere, and that actually forms the basis of a very nice summer high school intern project. So we get summer students high school students every summer. They spend about 6 weeks with us. What we do is send them down to the beach to collect seawater, or to the marsh to collect marsh water, sometimes to the sewage farm to collect sewage. And what they do is take that water, simply spin it down, and then look at the enormous variety and diversity of the phage that is in that water. By some estimates, there are up to 1 billion phage in a milliliter of seawater in these microbial mats in the sea. So it's incredibly abundant. And you can see it's very diverse. It's hard to find any two phage that look identical. And this was done by a high school student a couple of years ago. But all the phage are similar. They have a head that's filled with nucleic acid, and they have a tail that's responsible for delivering that nucleic acid from the head into its host. So Gabe and Jack were interested in these essentially DNA packaging and delivering machines. And this is a nice, accurate picture of Gabe and Jack, because Gabe is slaving away collecting data, and Jack is lying back having fun in his office admiring the data coming in. So both of these consist of a phage head, and then in this case a very long flexible tail, and in this case a phage head and a very short stumpy tail. It's coming straight out at us there, it's off to the side there, so this is a short stumpy tail. If we just look at the head, we can take advantage of the 60-fold symmetry and do a lot of averaging to get to quite a decent resolution. And we can look at the way these subunits are put together to form this very rigid capsid, which has extremely thin walls, and yet must withstand the pressure of the DNA packed inside it, which some estimates say is 10 times the pressure inside a champagne bottle. So evolution has come up with all kinds of exotic ways to build these capsid structures in order to withstand that pressure. But it's arguable that the more interesting part of this phage is really this working machine here, this tail machine. And in P22, which we're going to talk about, that tail machine is assembled from very few gene products, and it works to pump DNA into a pre-assembled procapsid, which sets off a cascade of other effects, including the maturation of the procapsid into a mature capsid, and some of these enzymes come off, some others come on, the proteins that have to interact with the host, and the whole thing is prepped for infection. One of the interesting things about P22 and other phage like it is that it uses a packing mechanism called head-full packaging. So this thing just keeps pumping DNA until the head is full, until it's under pressure. There's no signal that says stop pumping now. So this thing has to sense pressure inside the capsid head. So it's by its nature interesting. The problem with wanting to look at the tail is this lovely 60-fold symmetric thing, which gives us all that wonderful averaging to deal with noise, now becomes an asymmetric object. So it's much harder to collect the data because each one of these is 60 if you're doing the averaging, and it's 1 if you're not. So you need a lot of data. And that's where Gabe came in. He was completely indefatigable. He collected 5,000 images in no time at all. From that, collected 25,000 particles, and was able to come up with the structure of this now asymmetric object, which is the phage head with this large tail machine. It's certainly at much lower resolution than just the capsid, but it allows us to explore the makeup of this system, and we can look inside and we can see the DNA in layers, and we can look at the details of the tail machine. One of the things that was observed is that the DNA is very tightly wrapped around this portal, which led to the hypothesis that perhaps this is a mechanosensitive pressure switch, and that's how the phage is deciding, I am full of DNA and we have to stop pumping. That was one interesting part. So this tail machine is generally interesting. It's an enormous structure, 2 megadalton, 5 types of proteins, and 51 subunits altogether, and they're put together with various symmetry mismatches. We really want to know how this multitasking machine is assembled, but you can't really do it from a resolution like this. It's too low. So what Gabe did was look at purified phage tails all by themselves. Now there's many more of them per image, so we can collect more data, in this case 350,000 particles, and reconstruct that tail machine to a resolution of 9 angstroms. And now we're just starting to see some of the underlying structure and just starting to make out alpha helices. Nevertheless, you still can't figure out where one subunit stops and the next one starts, because we don't know how to fit this. There's no X-ray crystal structures of these. So what Gabe did then was look at expressed parts, an expressed region of this, the portal structure. And these are tiny little structures, now much smaller and much harder to see in the electron microscope. Nevertheless, 50,000 particles later, we had a structure of the portal. And this one was quite challenging because when it self-assembles, it assembles in different symmetry states. So those symmetry states all had to be sorted out before he could get that structure. So by this sort of divide and conquer approach, in the end, what you end up with is an understanding of how these different subunits are put together with various symmetry and asymmetry mismatches into this incredibly complex working machine, which has to multitask. It has to translocate DNA, it has to sense pressure, it has to interact with its host, and it has to eject DNA into its host. Gabe went on to do lots of other projects like that, and I'm happy to say that Jack and I just want to point out, we can fit there are a couple of X-ray crystal structures of parts of this available, and they fit in quite nicely, which is a good validation. And Jack continues to work with us to this day, and Dave Wiesler is now mostly resident in our group and doing all sorts of very beautiful work at increasingly high resolution. So that's the end of one story, and I'll tell another one to show a different value of these methods. And that was work we did with Jamie Williamson, first with a graduate student in our lab, Craig Yoshioka, who then handed the project over to Anke Mulder when he left, and she finished it off. And we were interested in looking at the dynamics of ribosome assembly. So ribosomes are self-assembling machines. And remarkably, in vitro, they can assemble just from their individual components. So if you mix RNA with the proteins, this thing will self-assemble. And in the case of the 30S ribosomal subunit, this structure will self-assemble from the RNA and the 20 proteins associated. So it's a very complex process. The RNA has to fold up at the same time as the proteins are binding. And until we did this work, the stuff that was known about this assembly process was sort of summarized in these so-called Nomura assembly maps. So they knew that the body, this orange part, assembled first, followed by the platform, and followed by the head in a 5' to 3' folding up. They also knew something about the rate at which these proteins were binding. These ones were binding first, and then these ones were binding later. And it was thought that there were probably parallel assembly pathways and there were probably assembly intermediates, but nobody had looked at this directly. All this data was coming from thermodynamics and kinetics on the ensemble. So we decided to look at this directly, doing so-called time-lapse EM. And in that case, we start with the RNA, we add the 20 purified proteins, mix those together to start a synchronous assembly process, and then monitor that over time. So we watch as the process assembles and look for the assembly intermediates. So at each time point of perhaps 10 time points, at each point we gather images of the assembly intermediates, and then we align and classify them and look for assembly intermediate populations. And we do that at various time points, usually 10 to 14 over a series of experiments. Once we have those assembly intermediates, these are them, we can look at them and sort them out in various ways. So here's one way of sorting them out. Over time, we can look at what's the most populated assembly intermediate to the least popular assembly intermediate. And we can put a sort of color map over that to emphasize that a little bit. And not surprisingly, what we see at the early time points are these very small assembly intermediates where the ribosomal subunit hasn't formed yet, and then later on a full ribosome as well as larger assembly intermediates. And early on, we see these small ones, we still see them later on in the process. So we can sort and search through these assembly intermediates in a whole bunch of different ways, and the result of that was to determine that there were at least 14 independent assembly intermediate states, and they were in 4 different groups. And to cut a very long story short, we worked this out by doing 3D difference mapping, by doing some subcomplex assembly, by looking at the X-ray structure, and sorting out which of the proteins belonged to all of these different assembly intermediates, and as a result of that, could put together this 3D structural assembly mechanism that correlated with the kinetic and thermodynamic data. And now we can watch the assembly of this ribosomal subunit from the start of the RNA, and then adding these proteins to form this group 1, and then going through a multiple parallel assembly pathways and reaching some dead ends to end up with the 30S ribosomal subunit. All of this work was done in vitro, and I'm happy to, first I want to say, this could not have been done without automation. In fact, I know Jamie asked several people to do this for years before we took it on, and everybody said, you must be crazy. And you would be crazy to do it without a high level of automation, because in doing these experiments, it was done 4 different times, each time with about 10 data points. And that final data set was done with 12 time points, 4,000 images were collected, 1 million particles were analyzed, and that was all done in 3 days. And then for the 3D data, 2,000 images were collected, 200,000 particles was done in 2 days. And you saw streaming past the hundreds and hundreds of 3D maps that had to be analyzed. And without the high level of automation to collect the data and this high level of automation to keep it organized, we wouldn't have taken it on. And I'm really happy to say that that work continues now. This was all done in vitro, and Deepa Sashitel, so postdoc working in Jamie's lab, is now approaching that from doing the work in vivo and looking at how cofactors are involved. I'll just briefly mention one other project that similarly did a very complicated sorting process. And this was done by Arnie Moeller, a postdoc in our group, together with Robert Kirchdorfer, a grad student in Ian Wilson's group. And Arnie and Robert met at Cabo a few years ago and decided they would look at the influenza virus replication machinery. This work's just been published in Science, and a commentary that accompanied it said, this seemingly impossible task was done. It's good that Arnie and Robert didn't know that when they took this on, but it was indeed an incredibly complicated task. These RMPs are very flexible and very heterogeneous. So it took an enormous amount of effort to sort them out, and they had to do it in pieces. They again used a divide and conquer approach. They reconstructed the middle part, this helical nuclear proteins that wrap around the RNA. They reconstructed the end separately. They reconstructed the polymerase end separately. They did the polymerase alone. They did gold binding studies, all to get an interpretation of what the structure looked like. But happily, even better than that, they got an interpretation of how this thing transcribes and replicates. By looking at this data and understanding what this machine was doing, they were able to hypothesize how it might be undergoing replication and transcription. So again, the real win, I think, of all of these structural studies is the dynamics that you get out of it, not simply the structure. Okay, so that's the good news. So what's the limitations? We're not happy yet by long chalk. You know, we've spent the last decade building this up, but there's so many things about it that we are not happy with yet. And what are the limitations? Well, in the sample side, we can only put one sample on a grid. It's very low throughput. We use 3 microliters of sample, which is an enormous amount of material, as you'll see, and the quality of the grids is usually pretty bad. On the data collection side, we can probably look at less than 10 samples a day, partly because we have to exchange samples. We're only collecting about 1,000 images a day, which is amazing compared to what we did 10 years ago, but still not nearly what we want to do. And the image quality is usually pretty poor. And then on the processing side, we have a bunch of wants and needs as well, but I won't have time to talk about those today. So what do we think we could enable if we could change this? If we could improve the grid quality and collect 10,000 images a day and improve the image quality at the same time, we should be able to get our 1 million to 10 million particles in a single day, and we should be able to do these atomic resolution structures in 1 day. If we could put multiple samples on a grid, improve the grid quality, and do 10,000 images a day, we could do 10 conditions a day, so that ribosome experiment could be done routinely in a day. And then if we could put 100 samples per grid and look at 10,000 images a day, we could do screening. And that would bring us much more in line with equivalent sort of hybrid methods. We want to be like the X-ray crystallographers. Once they have their crystal and they're at the synchrotron, they have their structure, and we'd like to be similar. SACs can do tens to hundreds of structures a day. We'd like to compete on that. And of course, light microscopy screening for X-ray crystals can do thousands of conditions a day, and we want to get to that point. So that's the goal that we've set ourselves for the next few years. So let's start with specimen preparation, because that's the trickiest part, and that's the sort of Achilles heel of everything. That has to be fixed before anything else can. So our goal there in specimen preparation is 10 to 100 samples on a single grid, using only 50 picoliters of sample and improving the grid quality. Our goal for image acquisition is 10,000 images a day instead of 1,000, and improving the image quality, and then we have similar goals for the streamlined processing. So we start with specimen preparation. And this is, as I explained, how we make cryospecimens now. We start with a bulk sample, put a bit on the grid, and we turn it into a thin film. The way we do that is we dump 3 microliters of sample, and we have these huge blotting pads which come and take most of the sample away. As a result, they turn 3 microliters into 3 nanoliters, and 99.9% of a sample ends up on the blotting paper in the bin, and only 0.1% of it ends up on your grid. That's bad because protein is precious and hard to make, and it's a tragedy for the really difficult proteins, like the membrane protein complexes, the eukaryotic complexes that have to be purified out of cells that can't be made up in other cells. So we really want to deal with that. The other problem that it's such a crude process, these two giant blotting pads coming down, that although there's 100,000 potential targets on this grid, most of them are not usable. We're lucky if we get 10% of the grid that has just the right sample thickness and spread that we need. So it's time to change that. It's notable that this is the one aspect of electron cryomicroscopy that hasn't changed in the last 30 years. We're still making grids exactly the same way, more or less, as we used to do 30 years ago, whereas everything else about cryo-electron microscopy has changed. The microscopes are better, the processing is better, the imaging is better, the cameras are better. So it's time for a change. And what we propose to do is to take this current method, where we have one sample per grid, 3 microliters, and very little usable area, and turn it using picoliter sample dispensing into 9 samples per grid, using only 30 picoliters and 100% usable area. And these are the factors that are involved. We would use 1,000 times less samples. So if you had 5 microliters of samples, this would last us forever. We could do as many experiments as we liked with it. Right now, if you have 5 microliters of sample, we can do 1 experiment and we're done. So that's crazy. This work was all enabled by a very talented mechanical engineer in the lab called Jain. So in order to do that, we need 2 new technologies. One of them is inkjet dispensing, which has become very routinely used in biology now with all of these DNA arrays. And the other is that we need new substrates, because we're not going to use blotting paper anymore. We have to rely on the sample spreading out across the grid by itself. So we designed a prototype. We call it Spot-It-On, because everything in the lab ends in on. And this is Spot-It-On. This is the very first prototype, and this is the design. We aspirate up a bit of the sample, and then we bring it up and approach this grid, the electron microscope grid. There's a camera that is looking at the spot as it's dispensed, so that we can know that we've successfully dispensed, and a strobe that flashes in synchrony with the spot. And there's a back camera that looks at the spot arriving on the grid, so we can control exactly where it arrives and how it's spreading. So once it gets there, it spots the grid on, we smash it into liquid ethane as usual, and then we can return the rest of the sample and save it for another day. So that's what it looks like in theory. This is what it looks like in practice. It's a bit of a mess, but it has all of those components, and they're exactly as we designed them. And here it is actually working. It's a little dark to see, but just to convince you that this thing does actually work, and it spots and it plunges. This is one of those 30 picoliter droplets flying through the air. We can watch it fly through the air. We can know exactly where it is in time, so we can do very fine timings on the system. This is 1,000 of the droplets being delivered. The strobe is strobing so that the spot looks stationary, but you can just see the 1,000 droplets accumulating. So we have precise control over the system. We can dispense between 1 and 1,000 droplets. We can put them also anywhere we like on the grid, as I'll indicate. So we can put a drop on any one of these grid squares if we like. And here's an example of that. This is what the grid looks like in the electron microscope, but this is what it looks like in the fluorescent microscope. And these are individual grid squares that have been delivered sample. And we can write clever notes to our advisors with that system and things like that. We also did another experiment where we put sample down on alternating grid squares. And if we look at that sample on alternating grid squares, we can see that's what we successfully did. This one doesn't, this one does, this one does, this one doesn't. There are 1,000 grid squares on a standard electron microscope grid. That means we could deliver 1,000 samples. If you only need a few pictures of each grid square, then we've got 1,000 samples delivered down onto the grid square. And if we can do 10,000 images a day, and you only need 10 images of each grid square, then we're looking at 1,000 samples a day. And now we're in that screening regime that the light microscopists are so happily in. So that's about where we are. We've built a second prototype. Now we're using these silicon nitride grids. This is what they look like. It's like a grid, but it's different design. This is silicon nitride. These are thin windows. There's 9 of them. And if we look at now we have 3 heads dispensing at the same time. So each head is dispensing 3 times across those windows. And if you look at the spots dispensing here, you can see them. Here's a very hydrophobic grid, so the spots are just sitting there, not spreading out. Here's a nice hydrophilic grid, and I think if you watch carefully, you'll see a spot arrive and just spread across the surface. And that's what we want, and that's about where we are. Here's an example of that. This was HK97, spotted with Spot-O-Tron into a silicon nitride window. Here's a huge, enormous area of vitreous ice, and if we go up in mag and go up in mag, here are our particles floating around in this enormous region of vitreous ice that is now controlled and and of the right thickness across. We have a ton of work to do on this. We are a long way away from making it successful, but we're extremely optimistic that this is going to really be the next breakthrough in getting control of our vitreous ice specimens and being able to do these large screening experiments and really compete with some of these other techniques. Okay. Oh, and this just shows you, nothing bad happened to these. We've done all kinds of experiments to show that, but this is the same sample done, in the literature some years ago by the standard classical methods. Okay, I'm just going to say a few words about our hopes for improving the throughput of the imaging. How do we do this imaging? This is what the electron microscope grid looks like in the microscope. So it's a bit of a mess. We have to find squares that are exactly the right, have the right thickness. These are too thick and some are ripped open, so we have to go target those. Then we zoom in. And we have to find, this is done over holy carbon, we have to find holes that are just right. We have to look for ones that have just the right ice, because not everything is. And we zoom in, and then we have to pick targets on those holes, and we do some focusing off sites because of the low dose requirements, and then we get these kinds of images. And we do that all day. This is a multi-scale targeting problem, and we do that all day, going through these multiple scale and collecting images, and we do 1,000 images a day. 1,000 is nice, but when you're looking at something like a rotavirus, and it's only four particles per frame, it's still pretty low, So we'd really like to address that, and our goal is 10,000 images a day, which we hope we can address partly driven by those new grid processes. If the ice was all good, and it was flat, and we didn't have to focus, and we didn't have to target, we wouldn't spend all of our time trying to get somewhere and waiting around once we got there, which is what we currently do. So that's our goal. But there's another very important goal, and that's related to what limits the resolution of our images. The theoretical number of subunits you need to average to reach atomic resolution was worked out many years ago by Richard Henderson and others, and it's in the thousands. We should be able to average a few thousand subunits together and get to atomic resolution. What happens in practice, here's two examples. This work was done in our lab for Tom Smith on cucumber necrosis virus, and this was done in Nico Gregorius' lab on rotavirus. And we look at the outcome of those reconstructions. There's the cucumber necrosis virus over here. We are at a resolution of about 5 to 6 angstroms, and we used about 4 million subunits to form this map. Here's the rotavirus. They're one of the highest resolution structures available right now, and you can trace the backbone, and this is under 4 angstroms, and Nico used 6 million subunits to get to that map. This one is better than that one for many reasons, partly because they have a nicer microscope than we do. But also, it's on film and not CCD. So in practice, the number of subunits we're using is millions instead of thousands. Why is that? Why do we need to average so many subunits? We have thought for a long time that it's because the specimen is drifting and the sample is being induced to move by the radiation. Here's a lovely illustration of what's happening. If your specimen is the wave which is moving and your particles are the surfer that is moving, both of these are probably happening while you're taking the image. And so all of the images we're acquiring are blurred to some extent, and we need to address that. Happily, and this brings me to the last story I want to tell, there's a brand new generation of cameras that's going to allow us both to improve the resolution, address these problems, and therefore improve the efficiency of data collection. And this work's been done by Angie Cheng in the lab and Melody Campbell, a graduate student who worked with Nico Gregoriev, who was a sabbatical visitor last year. And Scripps gets star billing here because thanks to their generosity, our group has one of the very first of these brand new state-of-the-art instruments, which we've been playing with very happily and producing all kinds of fun data. So what is this new generation of cameras? The difference between the old generation, the charge coupled detectors, and these new generation, which are the direct detectors, is that the charge coupled detectors have to turn electrons into photons. And they do that using a scintillator and fiber optics. Inevitably, when you turn an electron into a photon, you get the scatter, you get a large point spread function, you're losing resolution. The new generation of cameras detects the electrons directly. Here's an image taken with a CCD and this new camera, DDD. They look identical, but not if you look at the information that is transferred at high frequencies. In the CCD, you're losing information very rapidly. There's nothing left at 4 angstroms. On the DDD, you still have a nice signal at 4 angstroms. So if we hope to get to 4 angstroms and below, we have to have one of these devices. But that's not all. Even better than that, this generation of cameras actually doesn't record a single image. It records a movie in acquiring its image. So this is what one of the images looks like. But it's actually made up of individual frames that are read out at different doses. So if we add the frames together, we get that final image that we have. But each one is available to us. So if these frames are available, if these movies are available, we can watch beam-induced motion, and we can perhaps correct for it. And that's a project we did with Nico. So here's an example. There's 60 frames that make up this final image, and we're looking at each one of them. Each one is of exquisitely low dose, only 0.5 electrons per angstrom, so we can barely see these particles. So instead of looking at the individual frames, let's zoom in on a piece of this and look at the accumulating frames. Here they are accumulating, and you can see as they accumulate, we build up contrast. As we get dose, we build up contrast. So the detail emerges, but the particles also seem to be blurring as we build up. And let's look at 3 of them in more detail. You can see as we build up, okay, the particles emerging, and then by the end, it seems like it's streaking out, like the surfer, swooping down that wave. We aren't really seeing the detail. So let's see, are the particles in fact moving? We did this by looking at 10-frame averages and comparing the particles in each 10 frame to the next 10 frame to the next 10 frame, and tracking the particles over time. We had to do 10-frame averages because there's just not enough signal in a single frame. So if we do that, and then we look at what's happening, the edge of this carbon is staying perfectly stationary, and if you look at one of these particles, it's bouncing around like crazy. That particle's moving quite extremely, this one's moving quite a lot. If you actually quantify how much they're moving, you'll see that these are, lines are exaggerated for emphasis, but that's the scale bar, 25 angstroms. Some of these particles are moving by 25 angstroms while we're taking the image. To me, it's a miracle that we've ever gotten to 4 angstrom, if that is the chaos that is going on while we're taking the images. And that explains why we're using millions of particles to do the averaging, because most of them are blurred out. And of course, you can see a blur of 25 angstroms, but your eye cannot detect a blur of 5 angstroms or 10 angstroms. It would look invisible to you. Okay, so happily with that in mind then, if we can see that blurring, we can correct for the blur. So here's that 60-frame average with the blurred out particles with no alignment. We can translationally align them because we know where they are, we can track them, and we get a much crisper, sharper image. And that's a project that we did. Melody and Angie reconstructed rotavirus on these now translationally corrected images and got a structure like that. What does it compare like to the previous one? Here's this new structure from 2012 on the new cameras with motion corrector. Here's the old structure from 2008 on film. This one is still slightly better than this one, but this has 8,000 particles and ours has 800. We've reduced the number of particles we need by a factor of 10 by being able to cope with this effect. They also have a bigger microscope than us, so they have an unfair advantage. So that's where we are with that. We're enormously excited by this new generation of detectors. Everybody in the field thinks it's going to really dramatically change things. It's also going to be useful, and this is one quick mention of another project in the lab. Dimitri Liumpkis is a grad student working with Claudia Jozero, and they're looking at a very tiny particle called listerin. And what listerin does is associate with ribosomes and then mark these aberrant polypeptide chains that are stalled inside the ribosome and mark them for degradation by the proteasome. It's never been crystallized, and if you look at the particles, you see why. They are astonishingly flexible, and it's a little loose string, and it adopts all kinds of different forms. Dimitri developed an enormous pipeline of processing and was able to show that the structure adopts a continuum of states, flexing about 2 hinge regions, and using that was able to sort of hypothesize how this little machine might work in associating with the ribosomes and being able to do the sorry, I'm getting tired. The reason we're excited about the cameras again is that we will be able to do a thing called dose fractionation. We'll be able to take these little particles, dose them way up, so we get the contrast up, with those high-dose ones, figure out what their orientation is, and then use the low-dose set of movie frames to do the reconstruction. So we'll be able to get contrast as well as high resolution, and we have high hopes for that too. So I think that's all I basically have time for. There's many other stories that I wish I could tell. We worked with Bill Balch early on, looking at COP2 coats and cages. We've worked with Ian McRae on that lovely dicer machine and did some beautiful specimen labeling. Ian did some beautiful specimen labeling to help solve that problem. We've worked for many years with Liz, and recently her and Arnie did a great piece of work looking at how NSF behaves as a working machine. And for years we've worked with MG Finn. Most recently Melody did a project with him, and we'll certainly miss him very much. So all that remains to do then is acknowledge the most amazing group. We are privileged to have people like Angie Cheng working for us. Angie knows everything in the lab and contributes to every single project, and is the go-to person for everyone. Jim Polokas is a software developer extraordinaire. He came from us from Illinois to Scripps and has been with us the longest time. Tilak is a mechanical engineer who's developing the new Spotaton. Amber develops the processing pipeline. Jeff's a new addition and very welcome. John is our electron microscopy technician, and Sargis is a sysadmin wizard. We also have a great group of postdocs and grad students. Arnie Muller is our postdoc, and Dimitri and Melody are two grad students. Dimitri about to graduate, Melody in about second year. We're lucky enough, too, to have visitors. David is working with Jack, and Deepa is working with Jamie, but they spend most of the time in their lab to our great benefit. And for years, we've worked with Liz on and off, and she spends a half a day or a day working on projects for us at the moment. And then we're ably assisted by Lorraine, who is the maestro of workshop and party organization, and Judy Townsend. And Clint and I run the group. So my very final slide is just to acknowledge the great luck we have to be at Scripps. It's an extraordinary place. We have an amazing group. We have an amazing group of alumni who are now out in the world doing great things, and some of them come back. Happy to say that Gabe, after an extraordinary, even more astonishing postdoc at Berkeley, will be starting as an assistant professor here in a couple of weeks' time. We couldn't be happy about that. And some of these come in and out and visit all the time. And then, because we're at Scripps, we have access to the most astonishingly productive biological projects. And these are some, not all, of the PIs, and of course their groups, most importantly, that we've worked with over the last 10 years. And we're looking forward to even more fun and more exciting time over, hopefully, the next 10. Thank you so much for your attention.